Welcome back to this next video in which we are discussing valvular heart disease. Okay, so we're now going to continue on our discussion of pathological processes that can lead to valvular heart disease, and the next one in the list is myxomatous degeneration. So let me start by telling you what myxomatous degeneration is. But before we do that, let me start by drawing a picture of the 3 layers of connective tissue of a heart valve cusp, because we've long since lost our picture of that, and it's going to be very important for us to have that picture in front of us when we describe what myxomatous degeneration is. So we will see that the major valve that is often affected by myxomatous degeneration is the mitral valve. So I'm just going to draw a picture of the mitral valve again, up here. So this is the mitral valve again viewed from above as though we're in the left atrium. Here is the anterior cusp, and then the 3 scallops of the posterior cusp there. And now let us imagine that we're going to take a little portion out of here, and in fact I'll put a little box. Let's take a little box there, so we cut that little bit of the anterior cusp out, and then we're going to have a look at the different layers of connective tissue, and this is good revision for what we did earlier. So of course we'll start with the portion that faces the left atrium at the top, and we'll go down to the bit that faces the left ventricle at the bottom. So the first layer, of course, is an endothelial cell covering. So here's one endothelial cell, here's another endothelial cell, and of course the last bit also will be an endothelial cell covering. So down facing the left ventricle here, we'll have endothelial cells as well. And these will of course be sat on a basement membrane, so I'll get my red pen for this. So here's the basement membrane that they are sat on, like so. And now we're ready to draw the 3 different connective tissue layers that make up the heart valve cusp connective tissue. So remember, I'll just orient this. This is the left atrium here, and this is the left ventricle down here, and let me get the thinner pen back. So the connective tissue layer then that faces the left ventricle is the thickest one, remember, the fibrosa layer, the strongest, thickest layer of connective tissue consisting of collagen, that always faces the way that the blood is actually moving. So in this case the blood's going to move from the left atrium to the left ventricle in this direction, so the fibrosa is going to be towards the left ventricle. So I'll mark on the fibrosa first. So all of this layer here that I'm colouring in white, this is the fibrosa layer, and remember this consists of a lot of collagenous connective tissue with fibroblasts amongst the collagen fibres. Okay, the layer then on top of that is then going to be the layer that consists of glycosaminoglycans, also called mucopolysaccharides, and remember that's called the Spongy Osa Layer. So this here is the Spongy Osa Layer, and I'll remind you that that consists of those polysaccharides that we discussed earlier, which are called glycosaminoglycans, or GAGs for short. So glycosaminoglycans, or GAGs for short, GAGS. Alternatively, you can use that old name, which is to call them mucopolysaccharides, and it's actually going to be this layer, the Spongy Osa layer, that's going to become thicker in myxomatous degeneration, whilst the fibrosa is going to become degenerated, it's going to become less thick. And the myxomatous part of myxomatous degeneration refers to the fact that whilst you get degeneration of the fibrosa, you're going to get thickening of the spongiosa. So just completing up the picture then, the final layer up here in this greeny colour, that consists of elastic connective tissue fibres, and in the case of a mitral valve cusp, this is called the atrialis layer because it faces the left atrium. Okay, so that's just a reminder of the 3 different connective tissue layers of a heart valve cusp. So, I'll say it again even though I've just said it, myxomatous degeneration involves the atrophy of this layer. This layer is going to get much thinner, so I'll find the appropriate colour for this. So this layer is going to get thinner, so I'll draw it getting thinner, so imagine it getting much thinner now, and you have to understand this is the main layer that gives the heart valve cusp strength. It makes it rigid and solid, whereas this layer, the Spongy Osa layer, the layer that's going to become thicker, this is more a rubbery layer rather than a strong fibrous layer. So what's going to happen in myxomatous degeneration is the heart valve cusps are going to become more rubbery in nature, whilst the Spongy Osa layer, this is going to become thicker. So that's what myxomatous degeneration means for the heart valve cusps. The Spongy Osa gets thicker, whilst the fibrosa gets thinner, and this makes the heart valve cusps weaker. It makes them more rubbery in nature rather than solid, because the Spongy Osa layer consists of this funny connective tissue which has a more rubbery nature, whereas the fibrosa consisted of the solid collagen fibres. So this makes the heart valve cusps weaker. So the next thing to ask is, what actually leads to myxomatous degeneration? Well, the answer is, for the most part, we're not actually sure what leads to myxomatous degeneration. Most cases where you get myxomatous degeneration are what are known as sporadic cases of myxomatous degeneration. And that means that we don't actually know what has led to this form of degeneration in the heart valve cusps, why the Spongy Osa has got thicker and why the fibrosa has got thinner. Okay, so most cases are sporadic, and in these sporadic cases, the heart valve that is nearly always affected alone is the mitral valve. So in sporadic cases of myxomatous degeneration, valvular heart disease, it's nearly always the case that it's only the mitral valve. You can have sporadic cases, it's not unheard of having sporadic cases where more than just the mitral valve is involved, so maybe the aortic valve or the tricuspid or the pulmonary valve are involved as well, but nearly always, if you've got a sporadic case of myxomatous degeneration, it will just be affecting the mitral valve. And we'll talk about what the consequences of this are in a moment, but you can probably already predict what the consequences of myxomatous degeneration, the mitral valve, are going to be. Alternatively, myxomatous degeneration can be associated with a systemic, usually inherited connective tissue disorder. So it can be associated with systemic connective tissue disorders, and these are usually, as I say, inherited. They're usually genetic disorders, connective tissue disorders. And the big one that I'm going to talk about is Marfan Syndrome, a very, very famous systemic connective tissue disorder. So Marfan Syndrome is an example of a systemic connective tissue disorder that leads to symptoms all over the body, because connective tissue all over the body has got problems. So these systemic connective tissue disorders, such as Marfan Syndrome, can be associated with myxomatous degeneration. And when you have a systemic connective tissue disorder that's causing myxomatous degeneration of heart valve cusps, then it usually affects more than just the mitral valve. So these ones usually affect multiple heart valves. So if you've got myxomatous degeneration in multiple heart valves, you start to suspect that the person might have a systemic connective tissue disorder rather than just be a sporadic case. So multiple heart valves. Right, so we will come on to discussing Marfan Syndrome in just a little bit. But for now, let's go back and talk now about what the consequences of having myxomatous degeneration of the cusps of a heart valve are. And usually it's going to be the mitral valve in these sporadic cases. You can see it occurring in other valves if we're talking about a systemic connective tissue disorder cause, and even in the sporadic cases occasionally you can see it involving the other heart valves. But let's just focus on the mitral valve. So what's going to happen then is these heart valve cusps are becoming more rubbery, and what that means is that they lose their structural solidity. They become much more rubbery and much more flexible, and this means that when the left ventricle underneath contracts, what's actually going to happen is it's going to push the heart valve cusps backwards, and you're going to end up with regurgitation. And indeed, this is the result of myxomatous degeneration in any of the heart valves. So if you get myxomatous degeneration in any one of the four heart valves, what it leads to is that heart valve losing structural integrity and therefore being pushed backwards, getting inversion, of the heart valve after the chamber has relaxed. You'll get inversion of that heart valve. If we're talking, for instance, about the aortic valve, you'll get aortic regurgitation, allowing blood from the aorta to come back into the left ventricle. So it always leads to regurgitation of the heart valve. Now you might say, we're talking about the mitral valve here most of all, but the mitral valve has those chordae tendineae to stop it from inverting. So why are those not functioning properly? Well, actually what happens is the chordae tendineae are functioning properly, but you have to understand that the chordae tendineae are not everywhere. So let me just add this onto the picture there. So if I just draw on where the chordae tendineae might attach, remember you have two papillary muscles of the left ventricle. You have an anterolateral one and also a posteromedial one, like so. And what these will have is they'll have lots of chordae tendineae coming up, and of course these are underneath the heart valve. Remember, we are looking at the heart valve from above, so I'm imagining we can see them. We can't actually see them, they're underneath, they're in the left ventricle, and so are the chordae tendineae. But just for the basis of the picture, here are lots of chordae tendineae coming up from this papillary muscle, and this one also will have lots of chordae tendineae, like so. But the point is they don't go everywhere. There are gaps in between the chordae tendineae. So in between the chordae tendineae we have these spaces, and those are called the intercaudal spaces. So what actually happens is the cusp portion that's in between the chords, so the intercaudal portion of the cusp, that will actually invert backwards, so you get it pulling backwards. So in between the chordae tendineae you get that little bit flapping backwards. Now that would not normally happen in a healthy heart valve because it's got too much structural integrity to then pull back that tiny little bit. But when it becomes rubbery in nature, that bit can actually flap back despite the chordae tendineae being on either side of it. So what you effectively get is, if I view this from the side now, so let's imagine I'm a little man, so here I am, and I'm going to go into this space here and actually look at the chordae tendineae attaching from the side. So here's what we've got. Here is the heart valve cusp viewed from the side, and here are some chordae tendineae coming down here. This is the intercaudal portion of the cusp. What now actually happens is this balloons backwards like this when you get this myxomatous degeneration, and then the blood can sort of push its way through so it doesn't close properly and you do get regurgitation. So this is actually called interquartile ballooning, to give you the full name for it now. So you get interquartile ballooning, and this means that the heart valve cusp doesn't actually close properly and that blood can push up through these balloon regions and then go back up into the left atrium, and this is why it can lead to mitral regurgitation. And the same thing will be true if it occurs in the tricuspid valve, where of course we also have chordae tendineae, and if it occurs in the aortic or pulmonary valves where we don't have chordae tendineae, then this won't even be relevant. They'll just become very rubbery and floppy, and then they can just invert, whereas previously they wouldn't have been able to invert because they're too rigid. It causes regurgitation is the key thing to put here. So the heart valve affected becomes weaker, and this leads to that heart valve regurgitating. Okay, so to summarise what we've seen so far before we now move on to the discussion of Marfan syndrome, myxomatous degeneration is this not particularly well understood degeneration of the connective tissue of the heart valve cusp. You get degeneration of the fibrosa layer, which is the collagenous layer, which is the layer that's really important for giving the heart valve cusp structural rigidity, whilst you get an increase in the thickening of the Spongy Osa layer, which has a more rubbery nature to its connective tissue. So this makes the heart valve cusps, instead of being rigid, more rubbery, and therefore it leads to them becoming weaker and it leads to them regurgitating. Now, most cases of myxomatous degeneration are what we call sporadic cases, and in the sporadic cases it's nearly always only the mitral valve that is affected. However, there are examples where systemic connective tissue disorders such as Marfan syndrome will lead to myxomatous degeneration, and when this is the case, usually multiple heart valves are affected. And something I want to stress is that in systemic connective tissue disorders like Marfan syndrome, it is not 100% assured that you will get myxomatosis degeneration. It's actually quite rare for someone with Marfan syndrome to get myxomatosis degeneration. So it's not the case that if you have Marfan syndrome, you will get myxomatosis degeneration in multiple of your heart valves. It's the case that if you have Marfan syndrome and you are very unlucky, you will get myxomatosis degeneration in multiple heart valves. So what I now want to do is, and the bit that I wanted to stress is that we don't really understand the sporadic cases. We don't understand what causes this in most people. So now what I'm going to turn on to is discussing Marfan syndrome, what happens in Marfan syndrome, what we currently understand, and this will kind of give you a little bit of insight into maybe why myxomatosis degeneration occurs there. It's still not going to give you the complete answer. It's not going to give you the final answer of why does the Spongy Osa layer get thicker whilst this layer gets thinner, but it might give you a bit more understanding. Okay, so let's now discuss Marfan syndrome. So I've just put a little title here. So we're discussing Marfan's syndrome. So Marfan syndrome is a systemic connective tissue disorder. You're going to get problems with connective tissue all over the body, and it's caused by a problem with the production of the protein fibrillin-1. So fibrillin-1 is a polypeptide that is coded for by the gene FBN1. So the gene for fibrillin-1 is called FBN1, which is obviously just an abbreviation for fibrillin-1. And this gene is present on chromosome 15. Now, of course, you will have 2 homologous chromosome 15s in every single one of your cells, and each of these will have a gene for fibrillin-1 present on it. So just drawing this, so these that I'm now drawing in red here, these are representing the 2 chromosome 15s. The 2 parallel lines are just representing double-stranded DNA of the chromosome. And on each one of these, you will have a little region. Of course, it's completely out of scale, but it gets the point across. You'll have a little region which is the fibrillin-1 gene. So this is the fibrillin-1 gene on this chromosome 15, and this is the fibrillin-1 gene on this chromosome 15. And in someone without Marfan syndrome, that's the end of the story. Well, not quite, but you have 2 chromosome 15s, each with a healthy gene for fibrillin-1 on it, and both of them will produce fibrillin-1, and story is happy. In Marfan syndrome, that's not going to be the case. One of the genes is going to be dysfunctional in Marfan syndrome, but we'll discuss that again later on. For now, I just want to describe in a bit more detail what fibrillin-1 actually does. So fibrillin-1 is an extremely important component of elastic connective tissue fibers. So I promised you long ago, earlier on in this video, that we would discuss the structure of elastic connective tissue fibers in more detail, and here now is where we're going to do it, because it's really important for us to understand what fibrillin-1 actually does in the body, so for us to discuss the structure of elastic connective tissue fibers. So here we go. So an elastic connective tissue fiber, it is made up of 2 major polypeptides, namely elastin and fibrillin-1. So we're now talking about elastic fibers. And elastin polypeptides make up the core of an elastic fiber, whilst fibrillin-1 makes up the outer portion of the elastic fiber. And you have to understand that an elastic fiber is much, much bigger than a polypeptide. I mean, these things are getting to the size where you're just about able to see them on a microscope, whereas these tiny little polypeptides, you can't see those on a microscope. So loads of polypeptides come together to make an elastic fiber. It's the same when we're talking about collagen fibers. They are not just a single polypeptide, they are loads of polypeptides all stuck together. So an elastic fiber that makes up elastic connective tissue consists of loads of polypeptides stuck together, and it has a core region that consists of elastin polypeptides and an outer region which consists of fibrillin-1. So let me just draw this. So this is going to be the core of the elastic fiber. So this bit is the core, and this consists of loads of elastin polypeptides cross-linked together. So this is going to be the elastin polypeptide, and elastin's given a very nice name. You instantly know what elastin does just from its name. It's involved in the production of elastic fibres. So in the core, you're going to have loads of these elastin polypeptides. So here they are, all in this core like so. And they'll all be cross-linked together. So just drawing some of these cross-links on, these are the cross-links between the elastin polypeptides, like so. Okay, so that's the structure of the core of a elastic fibre. What then is around the outside? Well, this is where the fibrillin 1 comes in. And I'll now draw fibrillin 1 on this picture in red. So fibrillin 1 polypeptides are going to form what we call fibrillin 1 microfibrils, and then those microfibrils are going to be around the outside of the elastic fibre core. So how do you form a microfibril? Quite simply, you take loads of the polypeptides and you bind them all together, you cross-link them together. So if this represents a fibrillin 1 polypeptide, a single fibrillin 1 polypeptide, so a tiny little thing, you can put loads of these together, like so, and it will also go a little bit more ahead like this. So loads of fibrillin 1 polypeptides together will make what we call the fibrillin 1 microfibril. And if I now represent the fibrillin 1 microfibrils just as a rectangle, like this, the fibrillin 1 microfibrils are then put around the outside of the elastic fibre core, like so. So these are fibrillin 1 microfibrils in red here. So overall, you've got loads of elastin polypeptides bound together making up the core of the elastic fibre, and loads of fibrillin 1 polypeptides around the outside making up the outer region of the elastic tissue fibre. Okay, so that's where fibrillin 1 is really important in elastic connective tissue fibres.